Well, welcome to Chapter 16, the control of gene expression in prokaryotes, and this is going to be Part 1. So we've got your student learning objectives to get you started studying, and then here's a case study from a previous edition of the textbook, which you will find in PDF form in Blackboard along with the PowerPoint version of this lecture. So this is on stress, sex, and gene regulation in bacteria. And the focus is the Streptococcus pneumoniae, shown right here. This causes infection and death, and we talked about it back when we talked about the Griffith transformation principle during the Chapter 10 DNA structure. So Streptococcus pneumoniae can infect and kill mice, it can infect and kill us, and it is pictured over here. In 1940, penicillin, the first antibiotic, was discovered, and it was discovered serendipitously, as they say. Someone was a little sloppy with their sterile technique, and they left their plate open, and some spores, some fungal spores, landed on the plate, and they found a zone of inhibition, as they call it. And so penicillin is actually made by fungi and was used in treating World War II folk. So they didn't lose their legs as frequently, their limbs as frequently as people in World War I. But, you know, we got a whole bunch of other antibiotics after that. But now the problem we have, you know, we could cure this pneumonia and all kinds of other stuff, but now the problem we have is that these bacteria are multi-drug resistant. So they're resistant to multiple antibiotics. And I'm sure most, if not all of you, have heard of the growing antibiotic resistance of bacteria. One example is MRSA. MRSA stands for methicillin-resistant Staphylococcus aureus. And Staphylococcus aureus, of course, grows on our skin. It's a normal part of our microbiome, if you will. And but it can acquire resistance to a lot of antibiotics that we take, especially if antibiotics are taken and not completed. So you take part of your prescription, you feel better, and then you decide, I don't need this anymore. Well, that's when the bacteria can actually survive enough against that antibiotic that now next time you try to take that antibiotic, those bacteria that are infecting you will not be killed by that antibiotic. So it's important to follow your doctor's orders and finish the prescription. It's also important not to take antibiotics when you have a virus, when you have a viral infection, because antibiotics, as the name suggests, is against life. And bacteria cells are life. They're alive. They are cells. Whereas viruses, we don't consider those exactly alive. They certainly don't have cells. They don't have a membrane. So antibiotics do not work against viruses. Such as COVID-19, which we're facing, and other kinds of viruses like influenza. Streptococcus pneumoniae 19A is what we call a superbug. Superbug. It's resistant to most antibiotics. So, and I just talked about Griffith, and he discovered that bacteria can be transformed. And that means what? That means that they can be changed. So their phenotype can be changed. As you'll recall from that experiment, There were smooth and there were rough strains of bacteria, not strands. That's for DNA and RNA, but strains. S-T-R-A-I-N-S, strains. So there are two different strains of bacteria that are the name Streptococcus pneumoniae. One was rough and one was smooth. And the smooth one survived because it had a defense against the mouse immune system. The rough one could not. But we were able to change the phenotype, the traits of the rough bacteria into smooth bacteria by adding DNA. And so we know that transformation of DNA to acquire these traits does occur in nature. I had a colleague, I used to work at Elon as an adjunct for 2.5 years, and there was a colleague I had there who was taking her students and going to the local streams and finding out which bacteria out in the wild, if you will, had acquired antibiotic resistance. And they were finding quite a few. I mean, think about it. A lot of this, you urinate, it goes into our water system, and then out into the wild. So, and bacteria can uptake these DNAs that they develop through the process of evolution, and they can pass those on to other bacteria through the process of transformation as they die. But in order to take up this DNA, you have to be what is called competent or have competence. And in order to be competent to transform or uptake DNA into your cells, you have to have a set of genes called the COM regulon. The COM regulon comes for competence. This is a stretch of genes, 105 to 125 genes long, that is found in the bacterial DNA or the bacterial chromosome. And it is transcriptionally regulated. One of the most important pieces or genes or proteins that are expressed from those genes is called the competence stimulating peptide, or CSP. And CSP is going to allow the uptake of that DNA into the cell. So the thing about bacteria is, and going back toward the title of stress, sex, and gene regulation, they are under stress. They're under constant stress. They are single-cell organisms. They're fighting for their lives on a daily basis. They replicate every 20 minutes, which means that their DNA can be mutated every 20 minutes. So, because every time you go through DNA replication, you can have spontaneous mutations. So, stress, what is stress to them? How about some antibiotics? How about some penicillin or ampicillin or, I don't know, think of some other antibiotics. I can't think of any right now, but any kind of vancomycin, there you go. There's one. That's the antibiotic of last resort when all the others fail. So the bacteria are trying to survive, and they're trying to survive in this situation where we're putting them under extreme selective pressure to kill them because we want to live. And their response is can be to uptake these genes to adapt to the antibiotic environment we're exposing them to. And that's why you're going to get your nastiest infections in the places where you're using antibiotics the most, such as in hospitals. And those are called nosocomial infections. And you'll learn more about those in microbiology. But that's what's happening. They get stressed. They adapt because they're mutating every 20 minutes. And some of those mutations are going to allow them to survive these antibiotic pressures, and they're going to pass these genes on through things like sexduction, the conjugation that we talked about with rolling circle replication earlier in the semester. And then they can turn on these set of genes to allow the uptake of these DNAs out of their environment. So this chapter focuses on how the bacteria regulate these genes, these big, long what we call operons or regulons, like the COM regulon. So how are they regulated? The regulation of gene expression is critical for all organisms. And so we're going to talk about genes and regulatory elements, the level of gene regulation, what are the levels out there, not just in bacteria, but in all organisms, and DNA binding proteins and their role in regulating gene expression. We've already been talking about some DNA binding proteins with our transcription unit. Those included things like the TATA binding protein, it bound to DNA, and sigma factor, it also bound to DNA. So you have had an introduction into proteins that bind to DNA. So E. coli that lives in your gut, Escherichia coli, is the master of adaptation. And here's some rod-shaped bacteria that are E. coli shown here in this picture. So for example, you can have, they can be, well, they're living in your gut. And let's say you had, oh, I don't know, some lovely potato chips because you're stressing out, and you have nice salt in there, but they've also got lots of carbohydrates. And what are carbohydrates but chains of glucose? So they're polysaccharide chains. So you break them down, you got lots of glucose. What are you going to do with that? Well, E. coli is actually going to be able to use that glucose as a carbon source to generate cellular energy called ATP. And it can also use other kinds of sugars, lactose from your milk, arabinose, I don't know where that's from, maltose, I want to say, I don't know where that's from either, xylose. Anyway, there's all sorts of different sugars from your food. So when you eat different foods such as lactose that's in your dairy products, the bacteria in your gut are going to turn on genes to metabolize that sugar and use it as cellular energy. So they can adapt from one sugar to another. If amino acids are also absent in your gut, E. coli can solve that problem too. It can activate all the genes for production of amino acids, and there is a section of this chapter that talks about the tryptophan operon, but we are not going to go into that for this class. But the big conclusion is that we need gene regulation. It's critical for bacterial survival. So gene regulation is the mechanisms and systems that control the expression of genes. And we have all sorts of levels of gene regulation. So genes and regulatory elements, sorry that went a little blurry there. Structural genes are one type of gene, and when we're talking specifically in prokaryotes, we're going to be focusing on structural genes. They encode proteins involved in things like cell signaling, cell processes, and metabolism. An example is ribosomal subunits are made from structural genes. So the protein portion of your ribosome is made from a structural gene. Prokaryotes also have ribosomes, so they're going to have those structural genes as well. Then there's another variety of gene called the regulatory genes, and they encode products that interact with other DNA sequences. So they're going to be DNA binding proteins, and they affect the transcription and the translation of these sequences. So of course an example would be a transcription factor, such as the TATA binding protein, or for bacteria, the sigma subunit, or the sigma factor that binds the -10 and -35 consensus sequences in the promoter. You guys remember that? I know you do. You also have in your DNA these things called regulatory elements. Believe it or not, you already know what those are. We've talked about enhancers, we've talked about promoters. In prokaryotes, we're only going to have promoters, we're not going to have the enhancers, but these are DNA sequences. Elements or DNA sequences that are not transcribed, but they play a role in regulating the transcription of other nucleotide sequences. So went blurry there again. I've just got my title. We've got genes and regulatory elements continued. Now we've got a term called constitutive expression. And this just means that the gene is continuously expressed under normal cellular conditions. So what kind of genes might you always need to be expressed? Well, ribosomes. You are always going to need ribosomes in your cells. So those are going to be continuously expressed, or in other words, constitutively expressed. There's a term called that we say is positive control. This is when we stimulate gene expression. We turn transcription on. And you also can have negative control. This is when we're going to inhibit gene expression or turn it off. Just like you can turn a light switch on, or you can turn that light switch off. You can do the same with genes. Eukaryotic gene expression is similar to bacterial gene regulation, so we're going to look at that briefly. Levels of gene expression are shown here. Looks like it goes blurry every time. It's just got the title up at the top. Sorry about that. But we've got up top this compact DNA. So we know that as heterochromatin. And one step in a eukaryote, for example, is that we need to go from the heterochromatin up top to the next step down, which is relaxed DNA that we know as euchromatin. And we have yet to really discuss that process in our class, how that happens. And we'll be talking about that in chapter 21 with epigenetics. But we can go from compact DNA to relaxed DNA, and that's a level of control of expression known as chromatin modification. And I apparently did not spell modification correctly. Here, the next level is transcription. So we have been talking about transcriptional regulation in class, and you go from DNA to pre-messenger RNA if you're in a eukaryote. Then we have talked about the processes of mRNA processing for our last exam to get a messenger RNA. And then you are going to translate this protein. We talked about how RNAi can inhibit that from happening, at least temporarily. So that's another level of controlling gene expression. And then finally, you can have what are called post-translational modifications, where you're going to modify your protein to activate it, doing such things as what? As putting phosphates onto your protein, for example. That can modify it. We'll talk about other modifications when we get to epigenetics. So this chapter focuses on the transcriptional regulation because that's the level that prokaryotes or bacteria cells use the most. They use transcriptional regulation. So this involves transcription factor proteins that have DNA binding domains. And interestingly enough, prokaryotes regulate transcription using operons. So they've organized their genes into groups of genes called operons. So we're going to talk about these DNA binding proteins a little bit. So if you're a DNA binding protein, that means that you have part of your polypeptide chain is made up of amino acids. So if you imagine that we've got, I don't know, let's say we have an N terminus over here and a C terminus way over there. I hope you can imagine this. And we've got lots of amino acids in between. Let's say we have 300 amino acid polypeptide chain. And part of that 300, maybe a third of it, is a subset of amino acids that is responsible for binding to DNA. In other words, they have a function. They have a function, and that is to bind DNA, forming hydrogen bonds with that DNA. So they're not covalent bonds, they're hydrogen bonds. This is a temporary association. So they have a portion or domain of the whole polypeptide chain that is devoted to interacting with DNA. Within this domain are things called motifs. This is within the binding domain. It's a simple structure that fits into the major groove of DNA. Remember, DNA is made of minor and major grooves. And there are distinctive types of DNA binding proteins based on the motif that we find there. So, for example, we have these three, helix, turn, helix, shown in panel A on the left. Then we have the zinc finger. I think I'm going to change my pointer briefly to a red. It's easier to see on the white background. We have the zinc finger protein, and it's binding here to the major groove of the DNA. You can go back over here to the helix. We've got our pink helix. Then we've got a turn in yellow and a purple helix. So helix, turn, helix. And it's going to bind with another helix, turn, helix protein as what we call a protein dimer. The zinc finger protein is going to have zinc ions that help it bind to DNA, so it's going to have a cofactor. And then last, we've got this thing called a leucine zipper that has two helices that have a high concentration of the amino acid leucine. And when I was in graduate school, I took protein chemistry. My degree is really in microbiology and immunology, my PhD, but I took a protein chemistry class because I found it fascinating to learn about the different amino acids that when they're in a row like you find in certain proteins, you get this helical structure and why that is and what amino acids tend to give you that helical structure. So we have certain amino acids that give us these secondary structures. These secondary structure regions are within the DNA binding domain. So this is a motif called helix, turn, helix that's inside of what we call the DNA binding domain. The types of amino acids that tend to be in here, I'm going to switch back over to my yellow pointer because I'm on a darker background now. We've got positively charged amino acids such as K, which stands for lysine, R, which stands for arginine. Those are positively charged amino acids. And then these three, or at least these two, are polar. Glycine is not polar, it's nonpolar, so it might be part of these turns that aren't associated with the DNA. And they interact with the sugar phosphate and they form a hydrogen base pairing with the sugar phosphate backbone. Your phosphodester bonds, basically. So phosphates have a charge. And what is the charge on a phosphate? If you said negative, you were correct. So DNA has a net negative charge, and therefore positively charged amino acids would be attracted to that DNA. Again, they bind dynamically. This means that it's a temporary interaction, and these different transcription factors can actually compete for the same position on a piece of DNA. So this is a dynamic type of binding. These are a table that I expect you to memorize all of it. No, I'm just kidding. Just memorize some of it. What part? Let's go back. These are common DNA binding motifs. We just talked about helix, turn, helix, and zinc finger, and leucine zipper. The one that I want you to remember is this helix, turn, helix up top. And I am now attempting to switch to a pen. Will the pen show up when I circle this region? Oh, it's crazy, but it's working. Okay, so there we go. There is what you need to remember from this table. Helix, turn, helix are the only type of DNA binding motifs that we're going to find in bacterial cells. All the others, you can also find them in eukaryotic cells, but all the other types of motifs on this table are found not in bacterial cells, but in, the other type of cell, the eukaryotes. Go back into my highlighter pointer. All right, so we're going to go to the next slide. Your concept check. How do amino acids and DNA binding proteins interact with DNA? Do they, A, do it by forming covalent bonds with DNA bases, B, do it by forming hydrogen bonds with DNA bases, or C, by forming covalent bonds with the sugars? Let's see if I've got the right one circled. Yes, I do. It is this one in the middle. They form hydrogen bonds. If it were covalent bonds, it wouldn't be temporary. It wouldn't be dynamic. It would be permanent. So it's hydrogen bonds that are needed to create this interaction between the DNA binding protein and the piece of DNA that it's recognizing. All right, so moving on, outline of operons. In other words, how do we can control transcription in a bacterial cell? Well, we have to study how those genes are organized in the bacterial cell, and they're organized into what we call operons. There are mechanisms of negative and positive control. Inducible and repressible operons exist. We're going to focus on the lac operon from E. coli and lac mutations. Well, that's really going to be the next set of slides, but it's coming. So you've got this exciting animation, which I highly, highly recommend that you study. No, I don't. I'll just discard my ink. You need to study this a lot. I mean, honestly, this is how I learned it, by looking at this animation. So, I mean, I even had it in graduate school, but this animation was the key for me. So please study. Here it goes.
To utilize lactose as an energy source, E. coli must produce the enzymes necessary for its metabolism. The genes for these enzymes are adjacent to one another on the bacterial chromosome and are transcribed together. A group of bacterial genes that are transcribed together is called an operon. At one end of the lac operon is a set of structural genes, LAC-Z, LAC-Y, and LAC-A, that encode the enzymes necessary for metabolizing lactose. These three genes have a common promoter. The regulator gene, LAC-I, is located upstream of the structural genes. The LAC-I gene encodes a repressor protein and is controlled by its own promoter. This repressor protein may bind to a region of the operon called the operator. The operator overlaps the 3' end of the promoter and the 5' end of the LAC-Z gene. The lac operon is inducible, that is, the structural genes are expressed only in the presence of lactose. Active repressor proteins are encoded by the regulator gene. In the absence of lactose, these repressors bind to the operator and prevent RNA polymerase from initiating transcription. When lactose is present, it is readily converted into allolactose, which may then come in contact with the repressor protein. Allolactose binds to the repressor protein and causes a conformational change that prevents the repressor from binding to the operator region of the DNA. With the operator site open, RNA polymerase is able to bind to the operon promoter and transcribe the structural genes. The resulting mRNA is then translated, leading to the production of the 3 enzymes, beta-galactosidase, permease, and transacetylase, encoded by LAC-Z, LAC-Y, and LAC-A, respectively.
Oops. Can't see
Lactose
that.
is a carbohydrate found in milk. It can be metabolized by E. coli bacteria that reside in the mammalian gut. Because lactose is too large to diffuse across the bacterial membrane, it must be actively transported into the cell by permease, encoded by the LAC-Y gene.
Okay, so part of your ICE, you have to write down what those genes encoded. So LAC-Z encoded the beta-galactosidase. Enzyme protein, whatever, right here. LAC-Y encoded the permease protein that you're seeing here that's allowing lactose into the cell. And transacetylase was encoded by the LAC A gene. Make sure you've got those on your ice.
The lac operon. To utilize lactose as an energy source, E. coli bacteria use the enzyme beta-galactosidase, encoded by the LAC-Z gene of the lac operon, to break lactose down into glucose and galactose. The function in lactose metabolism of the third enzyme, transacetylase, also produced by the lac operon, is not yet known.
When you're filling out your ice, remember that beta-galactosidase does this. It's going to convert lactose into glucose and galactose, but it did something earlier in the video. I'm going to point that out in an upcoming slide in case you missed it.
Partial diploids have been used
we're
to
not
help
going here,
define
right?
the roles of the...
We're going to stop here for now. So we're going to go back to the PowerPoint slide. That's a whole can of worms, that partial diploid, and I'll need to draw that for you. Alright, so be working on your lac operon ice as we continue to go along. Operon structure includes what is an operon? It's defined as a group of bacterial structural genes that are transcribed together and the cis sequences that regulate the expression of these structural genes. So the cis sequences include the promoter and the operator. Operons regulate bacterial gene expression at the transcriptional level. So we're regulating transcription, not post-transcription, not translation, not chromatin modification, but transcription. So here's an operon. An operon, again, is a group of structural genes plus sequences that control the transcription. So you got structural genes that are downstream of the operator promoter. Gene A, B, and C for the lac operon. Those are LAC-Z, LAC-Y, and LAC-A. RNA polymerase is going to bind to this promoter unless something about the operator is going to prevent that, and that would have to be a protein that's binding there. But RNA polymerase is going to bind to this promoter, and we're going to initiate transcription. There's a separate location, not part of the operon. This is separate from the operon. It encodes a gene called the regulator, and it has its own promoter. It is also transcribed and translated into a protein that can bind to the operator. Okay, so this protein is going to be made elsewhere in the genome of the bacteria, and then it's going to move. It's going to be transported. It acts in trans from wherever it's been made to find the operator that it recognizes and binds to it. And that can affect whether or not RNA polymerase is going to do its job. But if RNA polymerase is doing its job, it's free to